Okay, today I'm going to be talking about Huntington's disease, and it's a disastrous mutation. I'm Malcolm Barry-kao. Okay, so today we'll be talking about adult onset Huntington's disease. There is another variety of Huntington's disease, which is early onset Huntington's disease, and that's primarily in children and makes up about 16% of Huntington disease cases, but we're focusing on adult Huntington's disease cases, as I said before. And so adult Huntington's disease cases are caused by a mutation in the HTT gene, which a gene is in a chromosome, which is in the nucleus of the cell. So in this HTT gene, which is a segment of DNA, codes for the HTT protein, and this mutation or this disease is caused by 35 plus repeats in the trinonucleotide CAG, and this is the mutation. It's an insertion mutation. So it's a progressive brain disorder, meaning that it gets worse and worse as time goes on. People develop it when they're 30 to 50, usually, as it progresses later on in life. And it is not prevalent within any particular population, meaning that all populations are affected evenly. No population gets snagged and kind of bears the brunt of this disease. And currently there is no cure. There's only treatments that mitigate the symptoms of the disease. As you can see on the right side, there's a normal brain, and then there's a brain affected by Huntington's disease. And you can see that the basal ganglia is basically all degraded away in the Huntington's disease picture. So, symptoms and mitigation. So, as I said, with the atrophy of the basal ganglia, you're going to definitely lead to brain problems, obviously. And so some of the symptoms are loss of memory, changes in speech, personality changes, disorientation and confusion, and notoriously in this disease, loss of motor control. This is like the staple of this disease, as it used to be called chorea, which means dance in ancient Greek, as people with this disease often have uncontrollable and sporadic movements, and maybe it does kind of look like a dance. Some mitigation, as I said before, there's no cure, but here are some treatments that can be used, including antipsychotics and antidepressants. HTT protein and lineage. So the HTT protein function is really unclear. Scientists don't really know much about it yet. However, it is found throughout the nervous system in both neuronal and non-neuronal cells, and it affects specific types of neurons, which is interesting more than others. And this disease is genetically passed on, and so it is autosomal dominant, meaning that it only takes one copy of this allele to cause a mutation for you to have the disease. And so therefore there's a 50% chance of passing it to your children, as you just have to luck out and the person who has Huntington's disease, that you don't get the allele that has Huntington's, and instead you get the other one. And what is interesting about this disease is that only there's cases that have no family history, and that makes up about 1 to 3%, and we don't exactly know how this happens. However, we do, we don't exactly know. And as you can see on this, the right side, you can see the effects of the toxic HTT protein, which is bigger and longer, and it leads to neuron degeneration. Okay, the impacts of mutation mitosis. So this mutation probably happens little by little, right? It's kind of progresses as on, and that's how it bypasses repair mechanisms and the checkpoints in the S phase of interphase and throughout interphase. And yeah, this occurs in interphase. And so it's a wrinkle mutation. It's kind of like a printer getting jam. The DNA polymerase really gets stuck, and it prints this codon several times. But this doesn't happen super often. It only happens occasionally. And this is how you can get more and more repeats. Okay, and most people have fewer than 27 CAG repeats, which means that you're at basically no risk at all of getting Huntington's disease. And on the right here, we can see a DNA polymerase making the other strand of the DNA there. Okay, so the impacts of mutation, transcription. So this is the first part of protein synthesis, and so mRNA, transcription, this is really the main start of it. DNA can't leave the nucleus, and it needs a messenger. It's just too big, and it's too long. So mRNA needs something short and sweet that can easily be exported out. So in this case, that's mRNA. And it's smaller and single-stranded, like I said. The transcription is similar to replication and occurs in the nucleus. So it kind of uses basically some of the similar enzymes here. So the helicase breaks the hydrogen bonds of the DNA and separates them in that image like that. And then it uses the leading strand and RNA polymerase starts to transcribe and make the mRNA, as you can see in the picture. And before that, the primer primes the DNA and stuff like that. But less is needed as it's using the leading strand, right? So it's faster. And the key thing is that mutation is passed on to mRNA. Even though the mRNA matures, the exons are cut out. Or the introns, sorry, are cut out. But yeah. So the impacts of mutation translation. So the second part of protein synthesis. And so the ribosome, which is made of rRNA, really directs this process. As you can see, that's that orange thing in the picture. So, and it directs the tRNA, which carry the amino acids, through translation using the mRNA template, this green strand here. And so each tRNA has an anticodon, as you can see this blue, and they have 3 of them. It's an anticodon is made of 3 nucleotide base sequences, which correspond to the complement base in RNA. Sorry, that says DNA, but I really mean mRNA. And each tRNA corresponds to a specific amino acid, as you can see on the top. That pink dot corresponds to a specific one, right? And so, just to say, ribosomes read the mRNA strand, they kind of translate it for the tRNA, and it directs the tRNA where to go. That's why it's called translation. And so the tRNA finds a new amino acid after it drops this one. And yeah, after that, the amino acid chain folds. And the key thing here is that the mutation is passed on to the HTT protein. And as you can see, here's the full picture. So there's normally more repeats on this normal side, as it's natural to have repeats of the CAG codon. However, there are definitely more codon repeats on the mutated side, and that's why I tried to have this contrast here. And so you can see that this is an insertion mutation, and there's a lot more CAG, GUC, and then glutamine amino acids on the mutated section, and that's what causes this toxic HTT protein. And here are some of the sources that are used. I hope you enjoyed this presentation, and see you later.